Good afternoon, everybody, and thank you for allowing me to give you a presentation by Zoom. Unfortunately, I can't be there, but I'm hoping this is a substitute, a slight one, that is. And today I want to talk about MSA and genetics and biomarkers. So my name's Henry Houlden. I work at the National Hospital for Neurology in London. I took over from Professor Quinn, who started an MSA clinic, and then I continued it once he retired, and we have a weekly clinic, an MSA Trust nurse, research fellow, and we routinely see many patients from around the country and bring them in, thoroughly investigate them, obtain the best diagnostic accuracy as we can, and then we're very forward in terms of genetics, biomarkers, and trials. So that's our setup in London. And of course, happy to see anybody there, but I only see NHS patients because I don't do private work, because I do research instead. So many of the treatments we're interested in MSA. We're obviously interested in the diagnostics and the management, and then we're very active in trying to treat and do as much as we can to support patients in terms of treatment for Parkinsonism, their blood pressure, bladder, impotence problems, mood, and then the trials themselves, such as exenatide and alpha-synuclein antisense oligos. This is a new trial therapy. So we try and enroll every patient we possibly can. Obviously, it depends on eligibility. Our typical MSA patients that we see in clinic, and I'm sure you're all familiar with this. So onset, 40s, 60s, very unusual over 70, age of onset. Ataxia, Parkinson's, rarely autonomic problems, bladder, blood pressure, emotional lability, and then quite rapid progression with minimal L-DOPA response. It's a classical patient of ours in terms of often wheelchair bound, bladder problems. He's got a catheter there. He's got ataxia in his upper limbs, and he's got dysarthria. Second patient.
And say it out loud.
Listen to this lady's voice, because patients with MSA get quite a distinct low volume but high pitch voice.
And
that lady's got a Caribbean accent, but we see it in
different
accents.
Can you say the French word for conjure? Look.
So you can see he had a very high pitched accent. And all of these patients, what is very interesting, many different backgrounds, but we do see the core features of MSA, which makes us think of a genetic disorder, a homogeneous disorder, because of all the many backgrounds, very, very similar features. And of course, when we look at patients' brains, pathologically very similar, and the scans at the top here are very similar when we look at MSA patients. Sometimes we get atypical patients. And this is a patient of Professor Lynch in Ireland, who's got a very asymmetrical progression, who had MSA. So as you can see, the right hand is not too bad. Left hand, very stiff, slow, painful as well, sometimes in patients. And when you see this, you worry about other diagnosis, but it's important to pick up, and we regularly video patients. So the two main types of MSA is the MSA that present with ataxia, MSA-C, and they're in the middle here. We think about other causes of ataxia, but MSA-C is where this red circle is. Onset over 50, late onset ataxia. When we see patients, we try and exclude the other causes either clinically or genetically. And the other type of MSA is MSA-P, which is MSA Parkinsonian. And these are the patients that presents with Parkinson's. Often they're diagnosed with Parkinson's, and then they progress a lot faster and they don't respond to the typical treatments. And then we say to ourselves, well, this looks like it's MSA-P type. And often when we see patients in clinic, they may have some C or some P features. It helps us to give them these names just so we understand the needs of the patient. So the question is, why do we think MSA has a genetic risk? Well, clinically, very specific homogeneous phenotype, pathologically unique alpha-synuclein deposited in the cytoplasm. MSA families, often MSA patients have a family history of Parkinson's. And when we go down, some of the Parkinson's risks are in MSA. And although the heritability is low, so it doesn't run in families, any genetic risk we find could be modifiable. So it could be changeable as a potential treatment. So the families exist, they're very, very rare. But when we see this, we want to think about other MSA lookalikes. And the MSA genetics, so as I said, it's usually sporadic, no family history. But then certain genes have been discovered that account for a small percentage of MSA. And I'll go over these. These are CoQ2, GBA, tau. Then there's this RFC1, which is canvas. Some of the ataxia genes as well can cause this. And I'll go over our progress in a genome-wide study of MSA. So the CoQ2 in MSA, now this was a major paper in New England Journal of Medicine by Soji Suji's team. A very excellent piece of work. And what he primarily found was CoQ2 is a risk factor mainly for Far Eastern MSA, so MSA from China and Japan. When we looked at our patients of European descent, which was patients from UK, Europe, the States, we never saw an association here. Although we think that there is some part that CoQ2 plays in MSA, because when we looked at brain tissue, this little circle has moved over a little bit. When we looked at brain tissue, we found that MSA had a lower amount of CoQ2 in the brain. So there is something there that suggests CoQ2 has a role in sporadic MSA as well as in families with MSA. So it's likely to be a risk factor. Certainly in European families, we don't see it running in the family. Next group of disorders, we looked at these disorders that are repeat expansions. We call them STR, short tandem repeat expansions. One of these is Huntington's disease, as an example. And this is where you get an expansion, a repetition of CAGs, 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 or a different motif in different disorders. And these are quite prevalent. So recent ones have been this canvas, which is a large repetitive number of these AAGGGs, FGF14, GAA repeats, a recent one in the Far East called NID, which are GGCs. And so what we did is we wanted to see just what percentage and what amount of these repeats accounted for our MSA patients. And so we looked at all of our MSA patients. The typical canvas, what we call canvas, is cerebellar ataxia, vestibular, which basically is dizziness, and sensory neuropathy. Those are the three things. They also get a cough. That's the classical canvas phenotype. But we do know that some patients don't get all of those features. And this is a patient with canvas. And patients look very similar to MSA patients. It's a little bit slower in duration. And there's more frequently a family tree, a family history. So this is a lady from my colleague, Mary Riley's clinic. And as you can see, she's got ataxia. She's got abnormal jerky eye movements. She's ataxic on her legs and needs a stick as well. She gets double vision. And when we do this particular movement, we can see abnormalities of the eye. This is called VOR. And when I looked in all of our MSA patients, or should I say our research fellow did, we found, initially we found 3. We're now up to about 8 patients. So it's about 2% of patients have this. I don't know, just go back. 2% of patients have this. And
Can you say British Constitution?
Patients present in a typical way.
Lovely little lilies. Lovely little lilies. Okay.
That gentleman there I saw in clinic with my fellow, he's got MSAC. He's a little bit Parkinsonian of his face as well. And he didn't respond to L-DOPA, and he progressed quite rapidly. If we look at his finger nose, he's abnormal. And if we look at his gait, he's very unsteady on his gait, difficult turning, and he's already had falls and needs a wheelchair. And that was one of our MSA patients that we found this RFC1 expansion in. And we found typical imaging, DAT scan, MRI, and we found a number of other patients with this. So I think this is a red flag we look for in clinic. Whenever I see a patient with MSA, I screen for all of these rare gene variants because I want to obtain the most accurate diagnosis. And then when we look back, I'll show you 3 patients. 3 patients here that we looked at with southern blot. This was our initial 3. And then we found these top 4 plots show that this peak here, this peak and then tail, is typical of this RFC1 canvas. And when we look at a southern blot, control patients have a little band here. Patients with the expansion have bands up here. They either have 1 where they get an expansion from their mum and dad, or they have 2 where they get a different expansion from mum and dad. So that was RFC1. Another recent repeat expansion is called in FGF14. It's a type of spinocerebellar ataxia. And I'll play this video. Patients are generally very similar to the canvas patients, ataxic on their legs, as you can see in this lady. Difficult turning. Often they have falls when they turn. They also have abnormal eye movements. You know, very jerky eyes with this nystagmus, and they get double vision. And these patients often present around about the same age as MSA, a little bit younger perhaps, usually mid-50s. And they present with more dizziness and balance problems before having a progressive ataxia. And the interesting thing about this type of ataxia, when we looked at families, we often didn't see a family history, because when we look at the inheritance, often when the patient gets this expansion from their father, the repeat contracts, gets smaller. So the inheritance is skewed, whereas if they get it from their mother, the repeat expands. So this makes it quite difficult to draw a family tree, because sometimes when they get it from their mother, it's large and they get the disease. But perhaps when they get it from their father, they don't, and it skips a generation. So this can make family trees quite difficult. And the other thing that has been advanced is there is a mouse model of this. And the mouse model does get tremors and dystonic features as well. It gets this stiff features. And this is a normal mouse. As you can see, it almost gets very stiff and falls backwards. We didn't develop that mouse, another group developed that mouse, but having a model like that can be very helpful for treatments. And when we looked at the brain tissue of patients with this FGF14, they do have cerebellar atrophy. They lose the neurons in the balance center. This is a control patient. This is the affected patient. They lose Purkinje cells. And we see a decreased in expression of the FGF14 gene. So we think that's the mechanism. We looked at our MSA patients with FGF14, and we were surprised that we found a fair number of patients with it in the total numbers that we screened. So if we looked at we looked at clinically diagnosed, and we looked at pathologically diagnosed, and we found 7 patients with FGF14. And we think that that's certainly a risk factor and maybe causing the disease in a small percentage of patients. We're looking at 1.5% to 2% of patients. And when we look at the patients, again, we have these little plots that prove the expansion by having this tail here. It's a bit difficult to interpret sometimes. We have to do it many times to get a clear blot. But this was in typical MSA patients. And this is an MRI of one of the patients, and they have the hot cross bun sign. Some patients had donated their brains, and we found that patients with FGF14 expansions do have the classical inclusions in the cytoplasm, and they get the intranuclear fibrillary inclusions as well. So we're talking, I think they're the more prevalent genetic causes of MSA. They're rare, but they're prevalent. We screen for them all. We do see some Parkinson's genes that look like MSA, and these are patients with mutations in the alpha-synuclein gene. They're either point mutations or duplications. And this is the gene here at the top. You can see a number of positions of the mutation. When we look at patients' brains, we see typical MSA type features. And these patients are also quite severely affected as well, often don't respond to L-DOPA. So that's one called G51D. This is a Swedish family, A53E, where it's inherited. And again, we see similar pathology. Patients that present with this look more like Parkinson's, quite stiff and slow. This is one of the first families described as an Italian and Greek kindred. They usually respond to L-DOPA, and the disease is a bit slower than typical MSA, but the pathology there are overlaps, and there are similarities to MSA, as some get autonomic problems, although some do get cognitive problems, which is not really the case in MSA. And this is this triplication where you get more than one copy. And although patients look like Parkinson's in the first instance, that when we look at their brains, they do look typical of MSA. So I've gone through the genetic or the more prevalent genetic causes. And now we think about biomarkers. Can we use some of those genetic causes as a biomarker for common sporadic MSA? And here we've looked at one biomarker called neurofilament light, or NFL. And we found that, we do see this very much in patients. So we see this NFL, which is a marker of neurons dying in the brain. In controls, it's very low, but in MSA patients, we do see this increase in life. And that's very important because as we see this increase, we want trials to then stop it increasing and reverse it ideally. And this is Viorica Chelban, who works with me, who's done a great deal of work in this area in trials. And she's now spearheading our work on alpha-synuclein and alpha-synuclein seeding. So for example, we looked at skin, and we do see excess alpha-synuclein in both MSA and Parkinson's in the skin when we biopsy it. And we've gone on, and our next steps, and this is what we're applying to different funding bodies, to look at longitudinal analysis of MSA. So our clinic over the last 20 years has been very important for patients, but patients have also been very generous to us, giving their blood, their spinal fluid. And we've kept this longitudinally over many, many years, along with the imaging, along with the scans. Both Professor Quinn and myself, Professor Bhatia, have all collaborated on this. And so now we want to look at some of the newer markers, such as the synuclein seeding in MSA, to see whether we can create a step change in biomarkers and diagnosis. So we want to look at spinal fluid and serum. We want to look at urine, and we want to do skin biopsies. The microbiome is something that we want to do. We haven't done it as yet. We think that looking at the blood's going to be more important. Looking at feces in patients with MSA and Parkinson's is tricky because the packs are quite difficult unless you've got a good carer to help you out. It can be difficult, but it's something that we do want to look at. And then last of all, I'll talk about our genome-wide study of MSA. Initially, we started this roughly 10 years ago. We collaborated widely, mainly looked at European MSA, got over 1,000 cases. We tried to work with everybody possible because this is a relatively rare diagnosis. We worked with brain banks and with clinicians. And my previous research fellow found a number of important MSA associated genes, particularly the tau locus, so microtubule associated tau. But our numbers were still small. So it started with over 1,000, but once we've done quality control, removed the DNA that didn't call quite as well as before, it dropped to below 1,000. But now we've worked with brain banks more extensively. We have over 1,000 brains and nearly 1,500 clinical cases. And this is our next step. We're doing a genome-wide study as well as whole genome sequencing of MSA patients. And this is what we're currently doing. And this is Virika's main project at the moment, along with her synuclein seeding assay. So those two projects we feel are going to deliver the most for MSA. So just to sort of thank everybody, grateful to many people, many collaborators, many funders, and of course, patients that have kindly given us their blood and allowed their brains to be donated. We're grateful to them and grateful to defeatmsa Alliance for listening to me. So our future plans are looking at the genetics, genome-wide study, looking at seeding, looking at longitudinal biomarkers, and looking at trials. We always on the lookout for very interested, keen MSA clinical fellows that want to be an MSA specialist in the future, as well as basic science PhD students. We're very keen to do that, and we're very keen to partner with institutions and funders to help us achieve our goals here. So thank you very much again, and best wishes.